Welcome to the deep dive. You're here because you're curious and, well, you want to get well informed quickly and thoroughly. Think of us as your personal guides, ready to unpack complex topics and maybe reveal those aha moments you crave. Today, we're taking a deep dive into how scientists pull apart, analyze, and even re-engineer the very building blocks of life itself. Cells, proteins, and DNA. We'll be navigating a stack of sources, from classic scientific texts to really cutting-edge research to bring you the most important insights.
Yeah, we're going from the microscopic, really right down to the molecular level, exploring the ingenious tools and techniques that have truly transformed our understanding of biology. You'll discover how simple inventions led to profound breakthroughs, how we can now read and even rewrite life's code, and even how mathematics helps us unravel the cell's intricate dance.
That's a powerful vision from the microscopic to life's deepest code. So let's unpack this. Our journey begins not with a grand theory, perhaps, but with a surprising truth about scientific progress. Often, breakthroughs aren't just about new ideas, but about new technology. Take the entire field of cell biology, for instance. It pretty much sprang into existence because optical craftsmen learned to grind lenses of, well, sufficiently high quality. This seemingly simple innovation allowed figures like Hooke and van Leeuwenhoek to peer into a previously unseen cellular world where, as one of our sources puts it, tiny creatures could tumble and twirl in a droplet of water.
Absolutely. That technological leap in lens grinding was foundational. It revealed a hidden complexity we couldn't have imagined. And what we're seeing in the 21st century is kind of a similar explosion of information driven by entirely new methods for analyzing cells, proteins, DNA, RNA, all of it. We now have access to the complete molecular blueprints for hundreds of organisms, from mustard weeds to humans, and powerful techniques to decipher that information. The long-range goal really is nothing short of obtaining a complete understanding of what takes place inside a cell.
That's incredible, a complete understanding. Wow. But okay, one of the first practical challenges in studying cells is simply getting enough of them to work with, right? That's where cell culture comes in. And when we talk about studying cells in the lab, these terms in vitro and in vivo come up a lot. They can be a bit confusing sometimes, can't they?
They certainly can, yeah. In common usage, in vitro often means in glass, and it's contrasted with in vivo, meaning in the living organism.
Hmm.
But in biochemistry, we use them a little differently. In vitro refers to reactions in a test tube without living cells, while in vivo means reactions inside a living cell, even if that cell is, say, growing in a culture dish.
Ah, okay. That distinction's important. And cell culture itself has this fascinating origin story, doesn't it? Tracing back to a 1907 experiment designed to settle a major controversy in neurobiology, the neuronal doctrine.
Indeed, yeah, that experiment truly laid the groundwork for the whole cell culture revolution. Small pieces of spinal cord were placed in lymphatic fluid, and individual nerve cells were observed extending these long, thin filaments. This provided really strong support for the idea that each nerve fiber is an outgrowth of a single nerve cell, a critical insight.
So, okay, how do scientists go from like a piece of tissue from an organism to individual cells they can actually study in a dish? What's the process?
Typically, a tissue sample is treated with specialized enzymes.
Enzymes, like molecular scissors.
Exactly. They digest the proteins in the extracellular matrix, the stuff holding cells together, and you also use agents that disrupt cell adhesion. This allows the tissue to be gently, teased apart into single cells.
Right. And once they're separated, what do these cells need to actually grow and thrive outside the body? It can't be just water.
No, definitely not. Most tissue cells require a solid surface, usually a plastic culture dish. Some even need the dish to be coated with materials like polylysine or components of the extracellular matrix to help them proliferate or differentiate properly.
What's really remarkable, and you touched on this, is that even in this artificial culture environment, these cells often maintain their specialized properties. Fibroblasts still secrete collagen, muscle cells can fuse and, get this, contract spontaneously right there in the dish, and nerve cells can extend electrical excitable axons that form synapses. It's amazing.
It's quite striking how much of their original behavior is retained. And this even extends to very specialized cell types, like embryonic stem cells. These are pluripotent, meaning they can be directed to differentiate into a wide range of specific cell types. They can even assemble into miniature organs called organoids, which are incredibly powerful tools for analysis now.
Organoids, wow. Miniature organs in a dish.
Exactly. And even plant cells, too. They can be cultured from a small piece of tissue to form a sort of disorganized mass called a callus, from which an entire new plant can be regenerated.
Okay, so cells in culture retain their properties, but there's a significant catch, isn't there? You mentioned this briefly. Most of these cells, particularly from vertebrates, they eventually stop dividing and die. What's going on there? Why the limit?
You're right, there's a limit. Most vertebrate cells have a finite number of divisions in culture, a process called replicative cell senescence. This is often linked to the progressive shortening and uncapping of telomeres.
Telomeres, those caps on the ends of chromosomes.
Precisely. They're protective DNA sequences. Human somatic cells, for instance, typically stop dividing after maybe 25 to 40 divisions because they turn off the enzyme, telomerase, that normally maintains these telomeres. So their telomeres shorten with each division. Although sometimes, even if telomeres are maintained, cells can halt division due to something called culture shock from excessive stimulation. It activates a protective mechanism.
So if researchers need, a potentially endless supply of identical cells for big experiments, how do they get around this limitation? How do they create these immortal cell lines that divide indefinitely?
Well, for human cells, you can often prompt them to proliferate indefinitely by providing them with the gene that encodes the catalytic subunit of telomerase, kind of restarting that telomere maintenance. However, some cells require more. You might also need to inactivate those protective mechanisms we just mentioned, sometimes by introducing certain cancer-promoting genes, oncogenes.
Hmm.
Rodent cells are a bit different. They often don't turn off telomerase in the same way and can undergo spontaneous genetic changes that lead to immortalized cell lines more easily. And of course, cancer cells themselves are a very common source of these transformed cell lines. They have distinct properties like growing without needing to attach to a surface and proliferating to much higher densities.
So what are the implications of using these immortalized cell lines in research? What do we need to keep in mind when we see studies using them?
They're incredibly valuable, no doubt. They provide vast numbers of uniform cells that can be stored indefinitely in liquid nitrogen. Makes experiments highly reproducible. However, and this is crucial, it's important to remember that they nearly always differ in important ways from their normal counterparts in the tissues they came from. There are trade-offs. Despite this, they're indispensable for applications like producing monoclonal antibodies.
Ah, the antibody.
Yeah, which are now widely used as treatments for human diseases like certain cancers or autoimmune conditions. So very impactful despite the differences.
Okay, so we've learned how to isolate and grow cells, even make them immortal. Now let's dig deeper. What about the molecular machinery inside these cells? Purifying just one type of protein from the thousands present. That sounds like a formidable challenge. How do scientists even begin?
It absolutely is a challenge, or at least it used to be much more so. Recombinant DNA technology, which we'll get to, has enormously simplified it. But whether you start from engineered cells or natural tissues, the process usually begins with something called subcellular fractionation.
Fractionation, breaking it down.
Exactly, to reduce the overall complexity. Then you follow that with increasingly specific purification steps.
So the first step is breaking open the cells. How do you do that carefully without just destroying everything inside?
Right, you need a gentle touch, relatively speaking. Cells can be broken up in various ways: osmotic shock, ultrasonic vibration, and even grinding them in a specialized blender. If done carefully, this ruptures many membranes but leaves the larger organelles like nuclei, mitochondria, the Golgi apparatus, largely intact. You end up with this thick slurry called a homogenate.
A homogenate.
Okay.
And then to separate these different components, the nuclei from the mitochondria, et cetera, scientists use something like a preparative ultracentrifuge, right? How does that work?
Exactly. This instrument, developed back in the early 1940s, rotates these cell extracts at extremely high speeds. It separates components primarily by their size and density. The largest objects, like nuclei, they sediment first at lower speeds, then mitochondria, then smaller components like microsomes, and finally things like ribosomes at very, very high speeds.
So it's like sorting by weight and size using centrifugal force.
Pretty much. And for even finer separations, density gradient centrifugation is used. This can separate based on size and shape, or even subtle differences in buoyant density. That buoyant density method was so sensitive, it actually helped provide direct evidence for the semi-conservative replication of DNA in a classic 1957 experiment.
Wow, that's a direct link to a major discovery. The power of these tools becomes really evident when scientists use these isolated components to understand how the cell actually works, piece by piece.
Absolutely. Studies of organelles purified by centrifugation have contributed enormously to our understanding. Experiments on isolated mitochondria and chloroplasts, for example, definitively showed their central function in energy conversion. Couldn't have done that easily otherwise. Even more powerfully, highly concentrated cell extracts, like those from Xenopus laevis frog eggs, which are huge cells, have allowed scientists to reconstitute and decipher complex processes right there in a test tube.
Reconstitute, like rebuilding the process outside the cell.
Exactly. Things like DNA replication, RNA splicing, even muscle contraction. The ultimate goal, really, is to reconstitute every biological process in a purified, cell-free system. Understand it from the ground up.
So once you have your mix of cell components, maybe enriched for certain organelles, how do you then purify a specific protein from that complex mixture out of potentially thousands?
Proteins are most often fractionated using column chromatography. It's a workhorse technique. A mixture of proteins passes through a column containing a porous gel matrix, and different proteins are delayed or retarded to different extents based on how they interact with that matrix size, charge, things like that.
Okay, and our sources mention several types of chromatography like ion exchange, gel filtration, and affinity chromatography. What makes affinity chromatography so particularly powerful?
Affinity chromatography. Yeah. It's truly remarkable because it takes advantage of specific biological binding interactions. It's highly targeted. For example, if you link a specific substrate molecule, something an enzyme acts on to the matrix in the column. The enzyme that acts on it will bind with very high specificity, while most other proteins just flow through.
So it fishes out just the protein you want.
Essentially, yes. Or you can use antibodies designed to bind only to your target protein. Because of this high specificity, you can sometimes achieve, 1,000-fold, even 10,000-fold purifications in a single pass. It's incredibly efficient, a real game changer for protein isolation.
10,000-fold purification. That's impressive. So once a protein is purified, how do scientists actually identify it if its identity isn't already known? Or analyze its basic properties like size? What are the key tools for that?
Right, you need to characterize it. That's precisely where techniques like SDS, polyacrylamide gel electrophoresis, or SDS-PAGE come in. This method separates proteins primarily by their mass, by their size, giving you a basic profile.
Okay.
For even greater separation, you can use two-dimensional gel electrophoresis. This combines two different separation procedures. First, separation by intrinsic charge using something called isoelectric focusing, then separation by mass using SDS-PAGE. Yeah. This technique can resolve, up to 2,000 different proteins on a single gel. It can even distinguish proteins that differ by only a single charged amino acid.
2,000 proteins. That's incredible resolution.
It is. And once they're separated on the gel, you can detect specific proteins using Western blotting. This involves transferring the separated proteins to a membrane and then probing that membrane with labeled antibodies specific to your protein of interest.
Okay, so Western blotting confirms if your protein is there, but what if you have an unknown spot on the gel? How do you definitively identify that protein or even map subtle changes to it, like modifications?
That's where mass spectrometry becomes absolutely indispensable. Think of it as a hypersensitive molecular scale. It transforms a sample into individual charged molecules, separates them by their mass-to-charge ratio, and then detects them.
Weighing molecules, essentially.
Exactly. Techniques like Maldi-Toph identify proteins by matching the masses of their peptide fragments, little pieces of the protein, to genomic databases. It's like fingerprinting. Even more powerful is tandem mass spectrometry, or MSMS, often coupled with liquid chromatography, that's LC-MS. This allows you to directly determine the amino acid sequences of individual peptides. You can identify hundreds of thousands of proteins in complex mixtures and, crucially, map post-translational modifications like phosphorylation.
Phosphorylations, these little chemical tags that act like switches.
Precisely. They're vital for cell signaling, and mass spec lets us see where they are.
Now, given that most proteins don't work alone, they work in complexes or interact with other proteins to perform their function, how do scientists identify those interacting partners, the collaborators?
Right. Context is key. Co-immunoprecipitation, or co-IP, is a key method for this. The principle is pretty simple. If your target protein is tightly associated with another protein inside the cell, when you use an antibody to pull down or capture the target protein,
its partner comes along for the ride.
Exactly. His partner comes along for the ride and can then be identified, usually by mass spectrometry. This method is incredibly useful for identifying proteins that are part of a stable complex inside cells, or even those that interact only transiently.
Okay, that makes sense. Find one, find its friends. And finally, for understanding a protein's actual three-dimensional structure, the shape that really dictates its function, what are the gold standards? How do we see that shape?
The two primary methods have historically been X-ray crystallography and NMR spectroscopy. X-ray crystallography provides incredibly high-resolution 3D atomic models, but you need to be able to crystallize the protein first, which can be tricky.
Right. Getting crystals isn't always easy.
No. NMR spectroscopy, on the other hand, is performed in solution. It uses the magnetic properties of atomic nuclei to compute structures. It's particularly good for smaller proteins, and it can even monitor changes in protein structure in real time, like during folding or binding to another molecule.
And what about really big things? Big protein machines?
Yeah, for very large protein complexes that are hard to crystallize or just too big for NMR, cryo-electron microscopy, or cryo-EM, has become incredibly powerful. It provides a more straightforward approach for these large assemblies. Ultimately, though, even just knowing a protein's amino acid sequence often provides a crucial clue to its function, because proteins with similar sequences often fold into similar structures and have similar biochemical roles. Sequence homology is a powerful predictor.
Okay, so we've covered the complex world of isolating, analyzing, and seeing the structure of proteins. Now, let's switch gears to DNA. Our sources note that DNA used to be incredibly challenging to analyze, much harder than protein in some ways. But now, it's actually one of the easiest biological molecules to work with. What changed so dramatically?
Recombinant DNA technology. It has truly had a dramatic, almost unbelievable impact. What once took years, literally years, now takes hours or days. It's now possible to determine the entire nucleotide sequence of a bacterial genome in hours, or, get this, an individual human genome in less than a day.
Less than a day for a whole human genome.
Yes, and this technology allows scientists to easily isolate any gene they're interested in and produce large quantities of its product, whether that's RNA or protein, even for molecules that are only present in tiny amounts in living cells. This is precisely how many human pharmaceuticals, like insulin or growth hormone, are produced today, by cloning the human gene.
That's a profound shift, enabling so much. So, okay, if you want to isolate a specific gene from a very long DNA molecule, like a chromosome, how do you cut it out with such precision?
The solution really began with the discovery of restriction nucleases, or restriction enzymes. These are enzymes from bacteria that act like highly specific molecular scissors. They cut the DNA double helix only at specific, defined nucleotide sequences called recognition sites. Different enzymes recognize and cut different sites.
So you can choose your enzyme to cut where you want.
Very much. And some enzymes, like a famous one called E-core-II, create these short, single-strand overhangs called sticky ends. These sticky ends are crucial because they can base pair with complementary sticky ends on other DNA fragments, helping different DNA molecules join back together, essential for cloning.
Right, the sticky ends help paste things together. And once you've cut the DNA into these pieces, how do you then separate them and visualize them, see what you've got?
Again, gel electrophoresis is the workhorse. Similar to proteins, but usually with agarose gels for the larger DNA fragments. The DNA fragments migrate through the gel based on size when you apply an electric field. The DNA bands themselves are invisible unless stained, most commonly with a dye called ethidium bromide, which binds to DNA and fluoresces brightly under UV light.
Okay, so you can see the different sized fragments. Let's dive into DNA cloning now. What does that mean exactly in a molecular biology context? We hear the term cloning a lot.
Right, it has different meanings. In molecular biology, DNA cloning refers to making many, many identical copies, typically billions of a specific DNA molecule, often a particular gene. The classic way, the foundational method, is to insert the DNA fragment you're interested in into a circular bacterial plasmid.
Plasmid, that's like a little extra circle of DNA in bacteria.
Exactly. It's a self-replicating genetic element. When you join your gene into the plasmid, you create recombinant DNA. This recombinant plasmid is then introduced into bacteria, often E. coli. And as the bacterium multiplies, doubling every 20 minutes or so, it replicates the plasmid along with its own DNA. Millions, billions of times. This allows both amplification, making lots of copies, and purification of that specific DNA fragment.
Amplification and purification in one go.
Clever. I've also heard of DNA libraries. What are those, and how do they relate to this cloning process?
Good question. Often scientists want to work with an entire genome, like the whole human genome, but it's way too big to handle in one piece. So a DNA library is essentially a collection of cloned DNA fragments that together represent the entire genome. It's spread out over maybe tens of thousands or even millions of individual bacterial colonies, each colony containing a plasmid with a different piece of the genome.
So it's like a library where each book is a different gene or DNA fragment. That's a great analogy, yeah. You can also make a cDNA library. cDNA is made from messenger RNA, the mRNA templates used to make proteins. This means cDNA lacks the non-coding intron sequences found in genomic DNA clones.
Ah, so just the protein coding bits.
Mostly, yes, which makes cDNA libraries ideal if you're focused on analyzing the protein products directly or expressing the protein.
Okay, DNA cloning in bacteria was clearly revolutionary, foundational. But it sounds like making and screening those libraries could still be quite time-consuming. Was there a later breakthrough that allowed scientists to work with DNA even faster, maybe entirely outside of living cells?
Absolutely. Yeah. A massive one. That's where the polymerase chain reaction, or PCR, comes in. Invented in the 1980s, it utterly changed everything. Its true genius isn't just its sensitivity, although it's incredibly sensitive. It's how it transformed what was once a painstaking, expensive, multi-step process involving bacteria into something fast, cheap, and accessible.
Fast, cheap, and accessible. The trifecta.
Exactly. It fundamentally democratized molecular biology. Suddenly, labs everywhere could amplify specific DNA sequences. It unlocked applications we couldn't have imagined just a few years earlier. It can amplify any nucleotide sequence you want, selectively, and it's performed entirely in a test tube. Billions of copies in just a few hours.
Billions of copies in hours. That's a significant shift in scale and speed. What are the basic steps involved in PCR? How does it work?
Each PCR cycle has 3 key steps, repeated over and over. First, heating the sample to separate the 2 strands of the DNA double helix, denaturation. Second, cooling it down to allow specific short DNA primers designed by the researcher to anneal or bind to their complementary sequences on the separated strands. These primers flank the region you want to copy.
So the primers define the target.
Precisely. And the third step is DNA synthesis. A special heat-stable DNA polymerase enzyme extends from the primers, copying the template DNA. This whole cycle, heat, cool, synthesize, is typically repeated 20 to 30 times. And because the amount of target DNA doubles in each cycle, you get exponential amplification. 2 copies become 4, 4 become 8, and so on.
Exponential growth. That's how you get to billions so fast.
This sounds incredibly powerful. What are some of the most impactful real-world applications we see from PCR? Where has it made a difference?
Oh, countless places. PCR's incredible sensitivity means it can detect even a single DNA molecule. This makes it invaluable for diagnostic applications detecting infectious agents, for instance.
Like for COVID testing.
Exactly like that. Detecting the viral RNA after converting it to DNA. It's also crucial in forensics. Detecting trace amounts of DNA left at a crime scene. You can then amplify specific regions, like short tandem repeats, or STRs, which are highly variable between individuals, to obtain a DNA fingerprint.
Right, the DNA fingerprinting. And it's used in research constantly for everything from cloning genes to measuring gene expression. It's just fundamental now.
The ability to manipulate and amplify DNA has clearly had a huge impact. What about DNA sequencing actually reading the exact order of those A's, T's, C's, and G's? How has that technology evolved alongside amplification?
DNA sequencing has undergone an equally dramatic, maybe even more dramatic, revolution. The original dideoxy method, developed by Fred Sanger, was groundbreaking, Nobel Prize-winning work. But it was laborious. It involved running 4 separate reactions, using radioactivity, manually reading sequences off large gels. It took days or weeks for even short sequences.
It sounds painstaking.
It was. But starting in the late 1980s, automation came in, using fluorescent dyes instead of radioactivity and automated jet readers. That made it much faster and safer. Then, after 2005, things really took off with the arrival of next-generation sequencing, or NGS, methods. Techniques like Illumina sequencing work by sequencing billions of short DNA fragments simultaneously, in parallel, a totally different scale. Billions of fragments at once. So what does that mean for us as individuals or for the field of medicine? What did that enable?
It means the cost of sequencing a whole human genome has plummeted from over $1 billion for the first draft in 2003.
$1 billion.
Yes, to less than $1,000 today. And it takes less than a day. This unprecedented accessibility has allowed scientists to examine thousands, now millions, of individual human genomes. We can catalog genetic variation across populations, uncover mutations that increase disease risk, tailor treatments. We can even sequence the genomes of extinct species like Neanderthals or woolly mammoths, opening incredible new windows into evolutionary history.
From $1 billion to under 1,000 in 2 decades, that's truly mind-boggling progress. Are there even newer methods coming along? Third-generation sequencing? Yes, the innovation hasn't stopped. Third-generation sequencing methods are now capable of sequencing much longer DNA molecules, which helps with assembling genomes. One type is single-molecule real-time, or SMRT, sequencing. It literally watches a single DNA polymerase enzyme incorporate fluorescently tagged nucleotides in real time as it copies a DNA strand.
Watching DNA replication happen.
Essentially,
Yeah.
yes. And another really innovative approach is nanopore sequencing. This doesn't even require DNA synthesis. It passes a single strand of DNA through a tiny protein pore, like threading a needle. And it reads the changes in electric current as each different nucleotide A, T, C, or G passes through the pore. Each base disrupts the current differently.
That sounds like science fiction, reading DNA as it flows through a hole.
It's amazing technology. And a major advantage of nanopore sequencing is that it can be performed with portable, handheld instruments. This opens up exciting possibilities for DNA and RNA analysis directly in the field, for tracking epidemics or environmental monitoring.
Okay, so we can sequence DNA incredibly fast, incredibly cheaply, even out in the field. But then you have these incredibly long strings of A's, T's, C's, and G's. How do we make sense of it all? How do we find the actual genes or understand what the non-gene parts do?
Right, getting the sequence is only the first step. That's where genome annotation comes in. Annotation is the process of marking out all the important features in the genome sequence, finding the genes, both the protein-coding ones and the non-coding RNA genes, and trying to ascribe a potential function or role to each one. It also involves identifying regulatory sequences, like promoters and enhancers, that control when and where genes are turned on.
How do we even find a gene in that long string?
One common way to find protein coding regions is to look for long, open reading frames, or ORFs. These are stretches of DNA sequence that start with a start codon and continue for a long time without hitting a stop codon. That suggests a protein could potentially be encoded there. Then you compare those potential protein sequences to known proteins in databases using bioinformatics tools. If it looks similar to a known enzyme, for example, that gives you a clue about its function.
But even with all this sequencing and annotation, do we now know what all the genes do? Have we solved the puzzle of the genome?
Oh, not at all. Far from it. For many organisms, even well-studied ones like yeast or humans, approximately one-third of the proteins encoded by a sequenced genome don't clearly resemble any protein whose function has been determined biochemically. We call them proteins of unknown function.
1/3. Still unknown.
Roughly, yes. This observation underscores a key limitation. While comparing genomes, comparative genomics, reveals a lot about evolutionary relationships and potential functions. It often doesn't immediately tell us how these genes actually function inside the cell or what specific roles they play in an organism's life. For instance, looking at the genome sequence alone doesn't really explain how this incredibly radiation-resistant bacterium, Dinococcus radiodurans, survives radiation doses that would shatter glass and kill us instantly.
Right, the sequence doesn't tell the whole story.
No. Further biochemical and genetic studies actually testing what happens when you change the gene are still absolutely required to truly understand function.
That's fascinating. So we can read the book of life, but we still don't understand every word, maybe not even every chapter. Given that, how do scientists actually figure out a gene's biological function in a living organism, beyond just looking at its sequence or comparing it to others? How do they test it?
Right, the functional testing. One of the most direct and powerful ways is to see what happens when that gene is missing or altered. This is the core principle of genetics. In the classical approach, going back to Gregor Mendel and Thomas Hunt Morgan, you'd find organisms, maybe fruit flies, with interesting appearances, white eyes instead of red, maybe weird wings. Then you work backward through breeding experiments to find the gene responsible for that trait.
Find the mutant, then find the gene.
Exactly. To speed this up today, scientists can intentionally create mutations. They might use chemicals or radiation to cause random changes in the DNA. Or they can use a technique called insertional mutagenesis, where pieces of known DNA are randomly inserted into the genome. If one lands in a gene, it usually disrupts its function, and you know which gene it hit.
Okay, but what about genes that are essential for survival? If you mutate one of those, the organism might just die, making it hard to study. How do scientists handle that?
That's a really important point. They use something called conditional mutations. These are very clever. These mutants function normally under certain conditions, called the permissive conditions, but they show the abnormal gene function under different restrictive conditions.
So you can switch the mutation on or off.
Essentially, yes. A classic example is a temperature-sensitive mutation. The protein made by the mutant gene might work fine at, say, 25 degrees Celsius, but it misfolds and stops working at 37 degrees Celsius. This allows researchers to grow the organism normally, then switch to the restrictive temperature to see what happens when the gene function is lost. You can study essential processes without immediately killing the organism.
That's ingenious.
Yeah.
And once you have a mutation, how do you figure out if it's causing a loss of the gene's normal function? Or perhaps maybe it's causing the gene to do something new or abnormal gain of function?
A standard genetic test for that is determining if the mutation is dominant or recessive. A loss-of-function mutation usually reduces or completely abolishes the gene's activity. These are typically recessive, meaning you need to inherit two mutant copies, one from each parent, to see the effect.
Because one good copy is usually enough.
Often, yes. A gain-of-function mutation, on the other hand, might increase the protein's activity, make it active at the wrong time or place, or give it a completely new function. These are usually dominant, meaning just one mutant copy is enough to cause an effect.
Okay, dominant versus recessive tells you about loss or gain. Now, what if you collect several different mutants that all show the same basic problem? Say, they all can't digest a certain sugar. How do you know if they are all mutations in the same gene, or if they're mutations in different genes that are all needed for that sugar digestion pathway?
That's where the complementation test comes in. It's a fundamental genetic tool. For recessive mutations, you mate two individuals, each homozygous, for one of the mutations, meaning they have two copies of that mutant gene. You then look at their offspring. If the offspring show a normal phenotype, if they can digest the sugar, it means the two mutations are in different genes,
Why? How does that work?
because each parent provides a working copy of the gene that was mutated in the other parent. The mutations complement each other.
Ah, okay. They rescue each other's defect.
Exactly. But if the offspring are still mutant, if they still can't digest the sugar, then the mutations are likely in the same gene. They fail to complement. This simple test has been used to figure out how many genes are involved in countless biological pathways. For example, it revealed that 5 different genes are required for yeast to digest the sugar galactose.
5 genes for one sugar. So you can identify the genes involved in a process using complementation, but how do you figure out the order in which they function in a biological pathway? Like step 1, step 2, step 3. That seems like a complex puzzle.
It can be, but there's a powerful genetic approach called epistasis analysis.
Pistis.
Yes. It basically looks at the effects of having mutations in 2 different genes at the same time, in a double mutant. If having a null mutation, the complete loss of function, in gene A, masks the effect of whatever is happening with gene B, meaning the double mutant looks just like the gene A single mutant, then gene A is said to be epistatic to gene B. This usually means gene A acts earlier in the pathway than gene B. Its defect prevents the pathway from even getting to the step where gene B acts.
Okay, so the earlier gene's effect overrides the later one.
That's the logic, yes. For instance, in the pathway where proteins are secreted from the cell, they move from the ER to the Golgi apparatus. If mutant A causes proteins to get stuck and accumulate in the ER, and mutant B causes them to get stuck in the Golgi. But in the double mutant A and B, the proteins still accumulate in the ER, then gene A must act before gene B in the pathway. The block in the ER prevents the proteins from ever reaching the Golgi where gene B would act. Epistasis helps map out the steps.
That's a really clever way to deduce order from mutant phenotypes. This is truly a powerful toolkit for discovery. Now let's talk about actually making these changes. How do scientists create these specifically altered genes or even engineer entire organisms to study them precisely? We're moving into genetic engineering here.
Right. Altered genes can be chemically synthesized from scratch now, believe it or not, especially shorter ones. Or, more commonly, they're constructed using recombinant DNA techniques, cutting and pasting pieces of DNA together in a test tube. Then, the challenge is getting this altered DNA into the cells or organism you want to study. This can be done in various ways. Microinjection, literally injecting DNA into a cell nucleus, using engineered viruses that carry the DNA in, or even something called particle bombardment.
Particle bombardment, that sounds intense.
It is. You coat tiny gold or tungsten beads with the DNA, and then you literally shoot them at high velocity into the cells, often plant cells, hoping the DNA gets through the cell wall and into the nucleus.
Shooting DNA into cells? Wow.
Once the altered DNA is inside the cell, the ideal outcome, especially for precise editing, is that it replaces the cell's normal copy of the gene through a process called homologous recombination.
Okay, you mentioned homologous recombination earlier. Could you just clarify for us again what that means in this context? Why is it so important for precise gene editing?
Yeah. Think of homologous recombination as the cell's own highly accurate find and replace system for DNA repair. If you introduce a piece of DNA into the cell that has sequences very similar or homologous to a specific region in the cell's own genome, like the gene you want to alter, the cell's machinery can recognize that similarity. It can then use the DNA you provided as a template to repair a break or replace the existing sequence. So it precisely swaps the original gene segment for the altered one you introduced. It's incredibly powerful for making targeted changes, not just random insertions.
Got it. So it's about harnessing the cell's natural repair system for precision. And this precise genetic engineering leads to what we call transgenic organisms, right? Organisms carrying foreign or altered DNA. How do we make, say, a knockout mouse where a specific gene has been completely inactivated? That seems like a big leap.
It was a huge leap, yes. Making a knockout mouse is a multi-step process. First, you create an altered version of your target gene in the lab, designed to disrupt its function. You introduce this altered DNA into cultured mouse embryonic stem, ES cells. These are special cells from early embryos that can develop into any cell type. You select for the rare ES cells where homologous recombination has occurred, meaning the altered gene has replaced one of the normal copies.
So you find the cells that made the swap.
Exactly. Then you take these altered ES cells and inject them into a very early mouse embryo, a blastocyst. This blastocyst is then implanted into a surrogate mother mouse. It develops into a chimeric mouse, a mouse whose body is a mixture of cells derived from the original embryo and cells derived from your engineered ES cells.
A mix of normal and altered cells.
Right. Some of these chimeric mice will hopefully have the altered gene present in their germline, cells the sperm or eggs. By breeding these chimeric mice, you can eventually produce offspring that inherit the altered gene from both parents, making them homozygous for the altered or knocked out gene. They completely lack the functional gene.
That's quite a process, but incredibly powerful.
Absolutely. This technology, developed in the 1980s, has been a major advance. It allows us to determine the functions of countless mouse genes by seeing what goes wrong when they're missing. We've seen effects ranging from developmental defects to things like premature aging in mice with a defective DNA helicase gene, directly linking specific genes to complex traits and diseases.
The ability to target and inactivate genes like that in mice was groundbreaking. But then came another revolution, something that made gene editing potentially faster, easier, and applicable to many more organisms. What was that?
That would definitely be CRISPR. CRISPR-Cas9, usually just called CRISPR. It was originally discovered as a kind of adaptive immune system in bacteria, a way they fight off viruses.
Bacterial immunity.
Yeah. But scientists realized it could be repurposed as a remarkably precise and versatile genome editing tool. It's revolutionized the field in the last decade or so. It works using a short guide RNA sequence that you design in the lab. This guide RNA directs a protein, usually Cas9, to a very specific target location on the genome sequence. Once there, the Cas9 protein acts like molecular scissors again, making a clean double-strand break in the DNA at that precise spot.
Okay, it cuts the DNA where you tell it to. What happens then?
The cell's natural DNA repair machinery kicks in to fix that break. Often the repair is imperfect, leading to small insertions or deletions that disrupt or knock out the gene. That's one way to use it. Or, if you provide an altered DNA template alongside the CRISPR system, the cell can use that template to repair the break via homologous recombination, precisely replacing the original sequence with your altered version.
So you can either knock out a gene or precisely edit it. What makes CRISPR so much more powerful and, well, revolutionary compared to earlier gene editing methods like the ones used for knockout mice?
Several things. Its main power lies in its simplicity and programmability. You can target Cas9 to potentially thousands of different positions across a genome simply by changing the sequence of that short guide RNA.
Just change the guide RNA.
Just change the guide RNA. You don't need to re-engineer the Cas9 protein itself for each new target, which was a major hurdle with older methods. And it gets better. You can use a modified, catalytically inactive Cas9 that binds to the target DNA but doesn't cut it. If you fuse this inactive Cas9 to other proteins that can activate or repress gene expression, you can actually turn specific genes on or off without altering the DNA sequence at all.
Wow, controlling genes without cutting them.
Exactly. And you can potentially do this for multiple genes simultaneously just by using multiple guide RNAs. Its adaptability and the ease with which it was exported from bacteria and shown to work in virtually all other experimental organisms, yeast, flies, worms, plants, fish, mice, human cells, that's what truly revolutionized the study of gene function so quickly.
So, with tools like CRISPR becoming widespread, we can now systematically alter genes on a genome-wide scale. What are some of the big insights coming from that kind of systematic approach? Are we learning things we couldn't before?
Yes, definitely. Extensive collaborative efforts have used these tools to produce comprehensive libraries of mutants for key model organisms, like yeast or cultured human cells. Libraries where every single gene has been systematically deleted or perhaps inactivated using CRISPR.
A library of mutants for every gene.
Pretty much. For example, there's a complete set of about 6,000 yeast mutants, each missing just one gene. And each mutant strain is bar-coded with a unique short DNA sequence tag. This allows scientists to grow large mixtures of all these mutants together under various conditions, different nutrients, stresses, drugs. Then, using sequencing to count the barcodes, they can rapidly identify which genes are essential for survival under those conditions or which genes affect growth rate. It's incredibly efficient for mapping gene function genome-wide.
That's powerful. What about organisms with fewer genes?
Studies using organisms like Mycoplasma genitalium, which has one of the smallest known genomes of any free-living organism, have been really informative. By systematically knocking out its genes, researchers identified that about 3/4 of its roughly 480 protein-coding genes are actually essential for life under laboratory conditions.
3/4 are essential.
Yes. And perhaps even more surprisingly, about 100 of these essential genes are of completely unknown function, even in this minimal organism.
Wow. Still so many unknowns, even for the essentials.
Exactly. It really suggests we still have much to discover about the basic molecular mechanisms required for life.
Another powerful tool that came along, often used for quickly testing gene function, especially in large-scale screens, is RNA interference, or RNAi. How does that work? Is it related to CRISPR?
It's different from CRISPR, but also very powerful for reducing gene expression. RNAi exploits a natural cellular mechanism that regulates gene activity. It involves introducing a double-stranded RNA molecule into the cell that has a sequence matching the messenger RNA, mRNA, of your target gene. This triggers a cellular pathway that specifically degrades that target mRNA, thereby reducing the amount of protein produced from that gene, effectively knocking down its expression.
So it targets the message, not the gene itself.
Exactly. It's frequently used in organisms like the fruit fly Drosophila and in mammalian cell cultures for high-throughput screening, where you might test thousands of RNAi molecules targeting different genes at once. And in organisms like the nematode worm C. elegans, it's incredibly easy. You can simply feed the worms bacteria that have been engineered to produce the double-stranded RNA targeting your gene of interest.
Feed them the RNAi. That's convenient.
It is. It allows for genome-wide genetic screens just by feeding. While RNAi is powerful, it's important to remember it often results in a knockdown, not a complete knockout like CRISPR can achieve. It also doesn't efficiently inactivate all genes, and sometimes it can have off-target effects, accidentally reducing the expression of other unintended genes. So results need careful validation.
Okay, so we have tools to knock out or knock down genes to see what happens. But beyond just knowing what a gene does, how do we go about seeing when and where it's actually expressed or active within a living organism, maybe even watching the protein product in real time?
Right. Understanding spatial and temporal patterns is crucial. Reporter genes are key for that. A common strategy is to take the regulatory region of your gene of interest, the part that controls when and where it's turned on, and fuse it to the coding sequence of a reporter protein. The most famous reporter is probably green fluorescent protein, or GFP, the molecule that makes jellyfish glow green.
Ah, GFP, I've heard of that.
Exactly. You essentially replace your gene's coding sequence with the GFP coding sequence under the control of your gene's promoter. When your gene would normally be turned on in a specific cell type or at a specific time, the cell produces GFP instead, and you can see those cells glow green under a fluorescence microscope.
So you see where the gene's switch is active.
Precisely. You can even fuse GFP directly to your protein of interest. This GFP fusion protein often behaves much like the normal protein, allowing you to track its location and movement over time within living cells.
Watching proteins move inside a living cell. Amazing. Are there other ways to see where genes are active?
Yes. Another important method is in situ hybridization. This technique directly visualizes the location of specific messenger RNAs, mRNAs, or non-coding RNAs within fixed tissues or cells. You use a labeled nucleic acid probe, a short piece of DNA or RNA that is complementary to the RNA sequence you're looking for. This probe hybridizes or binds specifically to that RNA wherever it's present in the cell. You can then visualize where the probe has bound, showing you exactly which cells are expressing that RNA. A big advantage is that this doesn't require any genetic engineering of the organism.
So you can look directly in tissues. And what about quantifying how much a gene is being expressed? Not just where, but the level.
For quantifying individual genes, the gold standard is usually quantitative RT-PCR, often called RT-qPCR.
Like PCR, but quantitative.
Exactly. First, you convert the RNA from your sample into complementary DNA, cDNA. using an enzyme called reverse transcriptase, RT.
Hmm.
Then you perform PCR on that cDNA using primers for your gene of interest. You monitor the amplification process in real time, often using fluorescent dyes. The rate at which the PCR product is generated is directly related to the initial amount of mRNA present in your sample. More starting mRNA means faster amplification.
Okay, so qPCR for single genes. What if you want a global snapshot, like the expression levels of all the genes in a cell at once?
For that global view, the dominant technology now is RNA-seq RNA sequencing. You basically isolate all the RNA molecules from your sample, convert them to cDNA, and then sequence millions or billions of these cDNA fragments using next-generation sequencing methods we talked about earlier. By counting how many sequence reads map back to each known gene, you get a quantitative measure of the relative abundance of essentially all RNAs present in the sample. It gives you a comprehensive expression profile.
A profile of everything being expressed. That leads to this intriguing idea mentioned in our sources, using guilt by association to figure out gene function. What does that mean in this context?
It's a powerful concept in genomics. The idea is that if sets of genes show similar expression patterns, meaning they are consistently turned on or off together under different conditions or in different cell types, they're likely to be functionally related.
They work together, so they're regulated together.
Often, yes. They might be a part of the same metabolic pathway, or components of the same protein complex, or respond to the same signal. Using computational methods like cluster analysis, we can group thousands of genes based on these shared expression patterns revealed by RNA-seq data. If a gene whose function we don't know consistently clusters together with a group of genes known to be involved in, say, DNA repair, that provides a strong clue, or hypothesis, that the unknown gene might also play a role in DNA repair. That's the guilt by association.
Finding function by looking at its friends.
Exactly. And this is being taken to an absolutely unprecedented level now with single-cell RNA-seq. Instead of profiling a whole tissue, you first dissociate the tissue into thousands of individual cells. Then you perform RNA-seq on each cell separately.
Sequencing RNA from single cells.
Yes. This allows us to categorize all the cells within a complex tissue into distinct groups based on their unique gene expression patterns. It's revealing incredible cellular diversity, identifying previously unknown cell types, tracking developmental lineages, and understanding cellular responses with amazing resolution.
That sounds like it generates massive amounts of data.
Oh, enormous amounts. Bioinformatics is crucial.
How about understanding the control mechanisms? How do we figure out how specific transcription regulators, those master switch proteins, actually control these gene expression patterns across the entire genome? Where do they bind?
A key technique for that is chromatin immunoprecipitation, usually shortened to CHI, followed by sequencing, so ChIP-seq.
Yeah.
It allows scientists to experimentally determine all the sites in the genome that a specific transcription regulator protein physically occupies in a living cell.
Find where the protein sits on the DNA.
Precisely. First, you treat the cells with chemicals that cross-link proteins to the DNA they are bound to, basically freezing them in place. Then you break the DNA into smaller fragments. Next, you use an antibody that specifically recognizes your transcription regulator of interest to immunoprecipitate or pull down that protein along with any DNA fragments it's cross-linked to.
So you fish out the protein and its bound DNA.
Exactly. You then reverse the cross-links to release the DNA fragments. Finally, you sequence these purified DNA fragments, ChIP-seq. By mapping these sequences back to the genome, you identify the precise locations where your transcription regulator was bound. Combining ChIP-seq data for regulators with RNA-seq data showing which genes are on or off allows you to build detailed maps of gene regulatory networks and identify the key regulators specifying expression patterns.
That is truly incredible, linking the regulator binding sites directly to gene activity. So, stepping back, what does all this incredible technological advancement from cloning to sequencing to CRISPR to ChIP-seq mean for human health and maybe beyond?
Oh, the impact on human health has been profound and continues to grow. As we mentioned, many essential pharmaceuticals like insulin, human growth hormone, blood clotting factors are now produced safely and abundantly by cloning the human genes into bacteria or cultured animal cells using recombinant DNA methods.
Right, making medicines.
Yes. And as genome sequencing becomes cheaper and faster, it's increasingly being integrated into clinical practice. It can be used to predict an individual's susceptibility to certain diseases, to predict how they might respond to specific drugs, pharmacogenomics, and even to guide personalized cancer treatments by identifying the specific mutations driving a tumor.
Personalized medicine based on our genomes.
That's the direction. Furthermore, transgenic animals, especially mice engineered using these techniques, can now be created to accurately mimic human diseases. This allows researchers to explore the molecular basis of these diseases in a controlled way and to efficiently screen for potential new drugs.
And it's not just human health, is it? You mentioned plants earlier. These technologies are changing agriculture too, right?
Absolutely. Transgenic plants are incredibly important both in research and agriculture. As we noted, you can take a single plant cell, modify its genes, and then regenerate a whole new plant from that single engineered cell.
Regenerating a whole plant from one cell, that's amazing.
It is. This allows scientists to engineer plants with improved traits. For example, improved nutrition. Golden rice is a famous example. It was engineered to produce beta-carotene, the precursor to vitamin A, in the rice grain itself. This aims to address severe vitamin A deficiency, which causes blindness and death in hundreds of thousands of children globally each year, particularly in regions where rice is a staple food.
Using genetic engineering for a major public health issue.
Exactly. Scientists can also engineer plants for resistance to pests or viruses, reducing the need for pesticides, or engineer them to tolerate harsh environmental conditions, like drought or salty soils, making agriculture more resilient in the face of climate change.
Okay, we've covered this amazing toolkit for observing, analyzing, and manipulating cells, proteins, and DNA. It's truly incredible. But our sources also highlight that understanding the cell's internal logic, how all these parts work together dynamically, often requires another powerful tool: mathematics. What gives math this, as one source puts it, almost magical power in biology?
That's a great question. Cell function and regulation, they depend on this complex web of transient interactions among thousands of different macromolecules, proteins, DNA, RNA. While schematic diagrams, arrows connecting boxes, are useful for visualizing relationships, getting a complete picture really demands a deeper, more quantitative understanding. We need to know precise terms about how molecules interact, how fast they catalyze reactions, and crucially, how their behaviors change over time. If a cartoon just shows protein A activates protein B, we can't truly judge its importance or dynamics without quantitative details, things like the concentrations of A and B, their binding affinity, their kinetic behaviors.
So you're saying it's not just if something happens, like A activating B, but how much and how fast it happens. The numbers matter.
Precisely. The numbers are critical for understanding the behavior of the system. Take a simple example: a transcription activator protein binding to a gene's promoter to turn that gene on. This binding interaction is reversible. There's an on rate, how fast it binds, and an off rate, how fast it falls off. These combine to define an equilibrium constant, often called K, which tells us the strength or affinity of the interaction. Math allows us to calculate precisely how much of the promoter DNA is bound by the activator at equilibrium, or what fraction is occupied. This fraction is often directly proportional to the gene's activity level. Okay, so math predicts the outcome based on affinity. Yes, and it can reveal non-intuitive things. For instance, depending on the starting conditions and the binding affinity, a 10-fold increase in the amount of activator protein might only cause, say, a 5.5-fold increase in the amount of bound promoter. It's not always a linear relationship. These kinds of quantitative insights are simply not accessible just by looking at the arrows in the diagram intuitively.
And if we want to know not just the final state, but how long it takes for the system to change or how it responds over time, that's where calculus and specifically differential equations come into play.
Exactly. Differential equations are the language of change over time. They describe how the concentration of a molecule changes moment by moment based on the rates at which it's being produced, its appearance, and the rates at which it's being degraded or consumed, its disappearance. They allow us to understand the transient dynamics of biochemical reactions, how the system behaves as it moves towards equilibrium, not just the equilibrium state itself.
Can you give an example of why dynamics matter?
Sure. Let's go back to that protein binding its promoter. Imagine we have two scenarios. In both, the final amount of bound protein at steady state is the same. But in scenario 1, both the on and off rates are slow. In scenario 2, both rates are much faster, but their ratio, which determines the steady state, is the same. The differential equations will show that even though the steady state is identical, the time it takes to reach that steady state can be significantly faster in the second scenario with the faster rates. This difference in response time can be critically important for how a cell reacts to signals. It's an insight you absolutely cannot get from a static diagram showing just the final equilibrium.
That's a key difference. So you've talked about individual interactions and how they change over time, but cells are incredibly complex networks, right? Thousands of interactions happening at once. How does math help us understand the bigger picture, the behavior of these interconnected systems?
That's where things get really interesting. Cell regulatory systems often employ recurring patterns of interconnection, sometimes called network motifs. These are like small, reusable circuit designs, larger modules of linked components that produce surprisingly complex and often biologically useful responses. Mathematics is indispensable in unveiling the properties of these motifs and understanding why evolution might have favored these particular circuit designs. They represent fundamental design principles of cellular regulation.
Network motifs, like common building blocks. Can you give us a particularly common example of one of these motifs and what it helps cells achieve?
A particularly common and very important motif is the negative feedback loop. This is where a component in a pathway, often the final product, ultimately acts back to inhibit an earlier step in its own production pathway, like a thermostat regulating temperature.
Product inhibits its own making. Makes sense.
Intuitively, you might think it just prevents the product from accumulating to excessively high levels. And it does do that. But mathematical modeling shows it does much more. Negative feedback also helps a system reach its final steady state faster after a change in input. And it makes the system more robust, meaning it dampens the system's response to fluctuations or noise in the input signal. It stabilizes things.
Faster, more stable.
Yes. But what's truly beautiful and maybe counterintuitive is when a negative feedback loop includes a significant time delay mechanism. Instead of just stabilizing the system at a steady level, a delayed negative feedback loop can actually induce sustained oscillations in the levels of the components.
Oscillations, like levels going up and down periodically.
Exactly, like a biochemical clock. These kinds of oscillations are crucial for timing biological processes, like the cell cycle or circadian rhythms. Math helps us understand how feedback and delays create these clocks.
So that's negative feedback stability, speed, robustness, or even oscillations with delay. What about the opposite? Positive feedback. What kind of behavior does that create?
Positive feedback is where a component stimulates its own production or activation, creating a self-reinforcing loop, like a snowball rolling downhill. This type of feedback can generate bistable systems.
Bistable, meaning two stable states.
Precisely. The system can exist stably in either a low state or a high state, like a toggle switch. Think of a light switch. It's stably either off or on. A transient input signal might not be enough to flip the switch, but once a strong enough push flips it to the on state, it tends to stay on even if the initial signal disappears, because of the self-reinforcement.
So it has memory.
In a sense, yes. This phenomenon is called hysteresis, where the current state of the system depends on its past history. Did it get pushed hard enough to flip? Biological switches like this are important for making irreversible decisions in development or cell fate choices. Mathematical modeling reveals that features like cooperative binding, where the regulator binds more tightly once some is already bound, are often important ingredients for generating these sharp, decisive, switch-like behaviors.
This raises another important question. As you said, cells are complex. Most genes aren't controlled by just one regulator, right? It's usually combination. How does math help us model and understand this combinatorial control by multiple regulators acting on the same gene?
That's right. Combinatorial control is the norm, allowing for much more nuanced and sophisticated regulation than simple on-off switches. We can model this mathematically by considering the probabilities of different regulators binding to the gene's regulatory region and how those combinations influence the gene's transcription rate. For instance, a gene might only be strongly active if, say, activator A is bound and repressor R is not bound. This is like implementing and not logic. Mathematical models can calculate the probability of this specific combination occurring, given the concentrations and binding affinities of A and R, allowing us to predict the gene's output under complex conditions involving multiple inputs.
So you can model complex logic gates. What about those feed-forward motifs mentioned in the sources? They sound quite sophisticated. What do they do?
They are sophisticated designs, yes. Feed-forward loops are three-node patterns where an input signal regulates a target gene, both directly and indirectly, through an intermediate regulator. There are different types. For example, an incoherent feed-forward loop might involve an input signal that quickly activates the target gene directly, but also slowly activates a repressor that then turns the target gene off.
Activate, then repress. What does that achieve?
Mathematical modeling predicts this arrangement can generate a brief pulse of gene expression in response to a sustained input signal. The output goes up quickly, but then comes back down even though the input stays high. useful for transient responses. Conversely, a coherent feed-forward loop might require the input signal to activate both the direct pathway and the indirect pathway through an intermediate activator before the target gene turns on strongly. This can act as a filter, ensuring the system only responds to prolonged or sustained input signals, effectively ignoring brief, noisy fluctuations. It ensures a costly cellular program isn't triggered accidentally by a fleeting signal. Math reveals the elegant logic in these network designs.
These models reveal really elegant design principles, but they often seem to assume that all cells in a population behave identically, perfectly following these mathematical rules. Is that truly the case in living systems? Are all cells the same?
Oh, no, not at all. And this is a really crucial point that's become increasingly clear. Cells, even genetically identical cells living in the exact same environment, often show considerable individuality in their responses. There's significant cell-to-cell variability.
Non-genetic variability, where does it come from?
It arises fundamentally because biochemical reactions inside cells involve molecules colliding randomly. It's inherently probabilistic or stochastic. Imagine a key regulatory protein present in only a few copies per cell. When that cell divides, it's unlikely that the two daughter cells will receive the exact same number of molecules just by chance. One might get slightly more, one slightly less.
Random partitioning.
Exactly. Even for proteins present in many copies, there are random fluctuations, noise, in the rates of gene transcription and translation over time. These random events mean that two identical cells can end up having different amounts of key proteins at any given moment, leading to different behaviors.
That's fascinating. So how do scientists actually study this inherent variability or noise between individual cells? How do you measure it?
They typically use techniques that allow measurements on single cells, rather than averaging over millions of cells. A common approach is using fluorescence microscopy to measure the amount of a fluorescent reporter protein, like GFP, fused to a protein of interest, inside individual living cells over time. Another powerful tool is flow cytometry, which can rapidly measure the fluorescence intensity and other properties of thousands or millions of individual cells as they flow past a laser beam. These single-cell measurements often reveal really striking diversity in gene expression levels or protein amounts across an apparently uniform population. Some cells might be on, some off, some in between.
So if there's all this randomness and noise, what kinds of mathematical models are needed to account for it? The simple deterministic equations aren't enough.
Deterministic models using ordinary differential equations are still incredibly useful. They're great for understanding the average behavior of a system or the underlying mechanism of a small circuit. But to really capture the effects of random fluctuations and predict the distribution of behaviors across a cell population, you need stochastic models. These models explicitly incorporate randomness, often simulating the probabilities of individual reaction events occurring over time. They predict not just the average outcome, but the probability of the system being in any particular state.
So they predict the variability itself.
Yes. Other modeling approaches are also used. Boolean networks, for example, simplify gene states to just on or off, allowing qualitative analysis of very complex gene regulatory networks where detailed quantitative data might be missing. And at the other extreme, there are agent-based simulations, which attempt to model the behavior and interactions of thousands or millions of individual molecules within a simulated cell volume, generating highly detailed, lifelike simulations that can capture spatial effects and stochasticity.
This all sounds incredibly complex, dealing with networks, feedback, randomness, variability, noise. How do biologists possibly draw meaningful, rigorous conclusions from experimental data that's inherently noisy and variable?
That's where statistics becomes absolutely essential. Statistics is, fundamentally, the mathematics of dealing with probabilistic processes and noisy datasets. It provides the framework and the tools to help us distinguish real biological effects from random experimental error or the inherent biological noise arising from that stochastic molecular behavior we just discussed. Statistics teaches us crucial concepts, like the importance of sample size. The more times we repeat our measurements, or the more cells we measure, the more confidence we can have in our conclusions, and the better we can estimate the true average behavior and the extent of variability. It provides the quantitative formulas and tests needed to move from looking at scattered, erratic individual data points to drawing rigorous conclusions about the key features of the data, allowing us to see the patterns and significance amidst the noise. It's indispensable for modern biology.
What a journey we've taken today, really, from the humble lens that first opened up the microscopic world to the incredibly sophisticated techniques we now have for isolating, manipulating, and sequencing life's fundamental molecules, DNA, proteins, all the way to these powerful mathematical models that are starting to reveal the intricate logic, the dynamics, and even the surprising variability of cellular behavior.
It's really quite something. These tools, from the surgical precision of CRISPR gene editing
Hmm.
to the broad genome-wide insights we get from single-cell RNA sequencing and the predictive power of these quantitative mathematical models. They are truly allowing us to dissect the elaborate molecular collaboration that gives rise to life. We're starting to see the hidden design principles, the network motifs, the feedback loops.
It's incredible to think how far we've come in understanding the cell, the fundamental unit of life. And yet, as we heard, there's still so much that remains to be discovered, those essential genes of unknown function. What stands out to you from today's deep dive? What's a key takeaway? Perhaps it's the idea that a single plant cell can regenerate a whole new organism, holding all that potential. Or maybe that a simple mathematical equation involving feedback and delay can explain why our cells oscillate like tiny clocks. As we continue to unlock the secrets of life, these technologies are clearly not just for understanding, but also for actively shaping our future, from developing new ways to treat diseases to finding sustainable ways to feed the world.
It really does raise an important, maybe even profound question, doesn't it? With these tools evolving so rapidly, what new unseen worlds will they reveal next? What fundamental biological processes might we discover or understand in completely new ways that could transform our assumptions yet again?
That's a powerful thought to leave you with. What's the next unseen world? Keep exploring, keep questioning, and join us next time for another deep dive.